Herc seat for this watch.
And my name is Erin Rainey, and I'm in the video engineer seat.
All right, thank you. And today we are exploring a seamount that is located approximately 60 nautical miles west of Poluana Island, which is part of the Sonsoran state. And it summits at a depth of 760 meters. And we plan to survey the seamount in order to collect the high-resolution imagery and physical samples around this area. So stay tuned as we slowly make our way up this seamount.
Oh yeah, eel. Who knows what kind of eel this is?
Do I?
No, it's not a cusk eel.
Cutthroat?
Yes, it's a cutthroat eel.
Nice.
Push in there.
Yep.
So, as I'm looking at this eel, to determine which genus it's in, I'm looking at the dorsal and anal fins and how they're aligned together. And if the dorsal starts before the anal fin, it's likely in the subgenus Iliophinae. And if it starts after, then it is Synaphorhynchus. So I think that one was a Synaphorhynchus.
Yeah, thanks for that, Megan, and shout out to one of our very loyal scientists ashore who's been supporting a lot of our fish records here. Over the last 3 weeks, I've been exploring the Palau National Marine Sanctuary. Dr. Ken Sulak, formerly from the U.S. Geological Survey, a blog to be coming out on our expedition website probably later today or tomorrow, just highlighting a little bit about some of the species, fish species that we've seen and the meanings that we have in both the Latin scientific names and the common names, and some interesting, curious facts. So just a little spoiler, but yeah, the Synaphorhynchus, the cutthroat eel, that also a common name is the short-finned cutthroat eel, describing the short fins. And the roots of this name, of the scientific name, refer to the gill openings, the fact that they're joined. Most likely is Synaphorhynchus affinis, the word they're related to. So yeah, thanks to Ken and many other scientists ashore that have been supporting this mission quite importantly over the last 3 weeks. We have a wide, diverse team of folks from all over the world joining us on shore. Thank you so much for all your support. We're sad to say that this is the last of the expedition dives of this Lebus Voyage 2. So we'll have about 14 hours of dive time remaining to explore this very interesting seamount that was marked for the first time last night and early in the morning. The plan is to cover some ground here. For those who are following on the dive plan, we basically landed just on north of the seamount, and then are going to be working ourselves up a kind of a ridge feature on the northern extent of the seamount, climbing gradually from our current depths of about 1,250 meters to the seamount summit at 775 meters. So a lot of vertical relief to go, quite steep topography here. And very interested to see what we'll see, and thank you all for joining us. It's been an absolutely joyful ride here, and can't wait to see what we see here in the next 4 hours together.
Excited, excited. Thank you. that, let's welcome our viewers online. We got some people in Colombia, Marshall Islands, Yokwe, Mexico, Netherlands, New Zealand, Australia, United Kingdom, Canada. We got some from Palau, Ali, and the rest from the US. Thank you for watching, and continue to watch with us as we move our way up this seamount.
Yeah, a couple of things here for those folks that have joined us recently, and courtesy of our previous watch here that took over after the ROV landed on the seamount. Yeah, the substrate is quite interesting. So they tried multiple times to collect samples, but all of these dark rocks that you see of them, they're all carbonate, all limestone corals that were once on the surface in the very distant past.
This is a jellyfish, right?
Yep, it's a jelly.
Okay. I'm just starting to like, is it a jellyfish or a sea cucumber? Because,
Oh yeah, there was that one that was really tricky.
yeah.
Yeah, also a little bit of recap from the previous watch in terms of now the substrate all being this carbonate that is covered by this black crust. Yeah, the sponge there on the right, these Tritopleura glass sponges seem to be dominant. Also a lot of pink corals or hemicoralliums, some acanthogorgias, and we'll see how we transition here as we go to different depths, and really now about to start going across a ridge feature. So we'll basically, for those that have seen the dive plan, we'll be going across two main ridges that really compose the main seamount. It's kind of a 90-degree system, I guess. Angle is the seamount is a bit of a sharp edge. So there's one ridge that will go up from the north to the top of the seamount, and there's a second seamount ridge that just intersects that in a 90-degree angle that is quite a bit sharper and steeper, and it goes for several kilometers at the top. And for those that joined us on yesterday's dive, that was truly, truly, truly remarkable in terms of the abundance of life, of all kinds of different taxa. I think that was the most diverse community I've seen on these sets of expeditions, and I believe that the same holds true for previous surveys in the Palau National Marine Sanctuary. And just highlighting that we're only just starting to scratch the surface of this tremendous reservoir of biodiversity that is the Palau National Marine Sanctuary. Looks like another
push in there.
cutthroat eel.
This is another one of those Synapobranchus, possibly affinis or calpii.
All right, come on.
So the dorsal fin is Is it?
Yep.
Yeah. So the dorsal fin on the cutthroat eel is the one that starts like almost more than halfway down its
Snap
body.
zoom there.
Yeah, so it depends on which eel, which species you're looking at. Um, if the dorsal fin starts before the anal fin, it's
Okay.
Iliophinae. If it starts after, it's Nephorhynchus, and like where on the body it starts after that in relationship to the anal fin is an indicator of species. So if they're more closely aligned with each other, the origins of the fins, then it could be Affinis or Calpii. If it starts really far back on the body, it's Brevidorsalis.
Oh, wow. Many different species.
Yeah, there are a number of different species. And it can be very difficult to tell them apart, especially since these animals are moving and these characters were based on specimens in hand. So characters that might be really obvious when you're holding a specimen aren't as obvious when the animal is alive and
Swimming away
swimming
from
away
us.
from you. Yeah, let's check out that crinoid. It's a bit different. So this looks like the Crotallometra that we've been seeing on a lot of things,
Push in there.
but it has these long filaments at the ends of the branches. So um, it's not that. This is different. It's really cool.
Yeah, beautiful imagery and good illustration here of the five-sided symmetry that characterizes these groups of invertebrates, the echinoderms, closely related to the sea stars, sand dollars, sea cucumbers, which those are more difficult to see the five-sided symmetry. You'd have to cut them in cross-section to see that. And yeah, those filaments look very interesting, but that's just the
Yeah, the long filaments at the ends are usually typical of the Xenometridae, but never seen one with just 5 feathers. And they have a lot of very fine cirri, those little walking legs. So it's very interesting, very distinctive.
yeah, thanks, Jake. That's
Okay.
great.
And come back wide.
It'd be cool if it swam for us. I love when they swim.
Have we seen a crinoid swim yet? On this voyage?
I don't know, not on our watch.
Maybe not.
Yeah.
That would be cool.
They're really cool when they swim.
It's very delicate and beautiful to watch a crinoid swim. I think there are some nice videos on Nautilus Live.
Yeah, I'll check it out.
Yeah.
Interesting object. Is that a
Fatty-
black
fatty
coral?
pathies.
Yeah, and is there a
And there's a sternostylus squat lobster on it.
Good eye.
Push in there.
That's great.
Okay. Come back one.
Yeah, definitely a black coral in the genus Bathypathes. And it looks like we are just about approaching waypoint 3 for those that are following on the expedition plan. So we've covered close to about 1 kilometer of a ambitious track we laid out. It would cover over 3 kilometers, so about 2.5 to go here to try to get as far up. And this dive track was designed to cross-sect a wide diversity of different depth ranges and habitat types. And we've been doing that systematically in all of our dives. This is the 6th dive that we've done on this second of Le Boost voyage. We did 7 on the first one. Each one of those, close to 100 hours of dive time on each one of the expeditions, so significantly increasing the amount of data we now have of the Palau National Marine Sanctuary, but still just really scratching the surface, and excited that this work will continue next year. Plans by NOAA's Office of Ocean Exploration and Research, our generous sponsors for these voyages, will be bringing their own vessel down here to Palau for a series of expeditions to continue mapping, characterizing, and exploring the Palau National Marine Sanctuary as part of our larger initiative to address a commitment made by the U.S. government at the 2022 Our
We're
Ocean
moving
Conference
155 now, Jake.
to support ocean mapping of the Palau National Marine Sanctuary, really building on long-standing collaborations between the government of the United States and Palau to work together on various issues, including climate change, environmental protection, and regional security. And for us, as part of the Ocean Exploration Trust, very proud to be part of this larger initiative. And thanking all of our viewers to have joined us on this endeavor, and hopefully you will continue joining us and partners as they continue exploring this exciting place that is the deep waters of Palau.
May, when you get the chance, I would love to get your thoughts on these rocks. Earlier I saw them kind of trying to get a sample, and it was just sort of soft. So I don't know if that's changed. When you get the chance.
So Lodak was just saying he would love to get your opinion on the rocks.
Yeah.
They were adjusting the
I was putting on another
adding more
layer.
layers.
Yeah, I think that we're looking at similar kinds of carbonate or sedimentary-based rocks that are covered in a thick manganese oxidized crust. That crust is also dusted with a layer of sediment, but if we zoom close, I'm sure we'd see that classic botryoidal texture that we've been seeing on previous dives. And these crusts are so thick that I think depending on where a piece is broken off, sometimes we can just end up with a piece of manganese oxide. The previous dive mentioned they had seen these kind of pillar-like features, and we did collect a rock a couple 100 meters downslope from where we are now. But it'll be exciting to collect a rock and maybe a little bit later in our shift when we get a little bit more upslope, see if we find a good candidate and see what it is. It's very, very interesting to be around these really, really old, geologically old features, because of course they create excellent habitat for these slow-growing and incredible corals and sponges that we've been seeing, but also you know, you would expect to see volcanic rock, but the processes at work here have been over millions of years, so that's why we're seeing this thick manganese oxide crust. And yeah, just really, really excited to hear from whatever geologists continue to work on these rocks in the future and continue to study the geologic history of this setting. It'll be really interesting to read the kind of papers that come out of that, since we have a really nice collection of specimens to work with.
Yep, thank you.
Yeah, thanks, May. And I think it might be good also for context for folks that might have not been joining us on previous dives to just point out we have desperately been trying to collect a lava sample on, on the
Yes.
200 hours that we've been now exploring. And I don't think if we've been successful yet.
Not in the south. In the north,
Yeah.
we do. We have definitely one or maybe two basalts that are for sure from the north. But on this Levy's Voyage 2, all of our rocks are, yeah, definitely sedimentary in origin, and a range of types of sedimentary rocks, from really loosely consolidated sediment that just crumbles in your fingers when you crack open the rock, to kind of conglomerate looking with a lot of little fossils, sort of carbonate rock, and then some more classic like limestone. So a lot of processes at play, and a lot of different timescales at play, because there's also great diversity in thickness of manganese oxide crust, from a couple millimeters to over 3 centimeters thick, which
Wow.
is really incredible to think about those, the slow growth of those crusts.
Yeah, thanks, May. So, yeah, I mean, it just putting it very in simple terms. So if you were to basically here drill into the seafloor, the very top would be that layer of crust, and that has accumulated over long time. And then as you go deeper into it, you would have limestone or calcium carbonate based on what we've collected in the past, and we have seen part piece of limestone inside. And that is coral reef that was once within the photic zone or the sunlight zone, and has now subsided and is now here in 1,200 meters. And then eventually, at some point, you would get to the basalt, but you'd have to drill down quite a bit. And so, yeah, that's very fascinating, is that you have different layers accumulating over time, and as if you dig deeper, if you will, onto these layers, that can uncover you and give you sense of how these places formed. Of course, many of these places that we're exploring, this seamount was not at this exact spot in the past, so they also moved across different areas. The seafloor is not a stationary thing. It moves. New seafloor is created at mid-ocean ridges, and is then destroyed at other places where it subsided. And that study is just a very fascinating study of geology, just to understand the time dimension of this. And I always think it's fascinating to think about, when we think about the history of our planet, 4.5 billion years, and the history of humans really only about 300,000 years, and how much we have done in those 300,000 years, when that is really a drop in the bucket of our planetary history.
So it looks like we did, we touched bottom at about 1,500 meters or so. So, oh, Is this a sea spider?
It sure is.
Whoa,
So
that is cool.
Now you push in there.
So those two green dots are 10 centimeters apart or 4 inches apart. So this animal is very large.
wow.
And it's got something. It must be feeding on an anemone or something.
It's like an amphipod.
Yeah.
Oh, there, it is. Oh, it's two amphipods that are living on it. I see. Normally these amphipods live together on stalks, but I guess the sea spider works very well as a stalk.
Much more mobile.
Yeah, that's cool. I've never seen this association before.
Very interesting.
It is very interesting.
What is the, or do we know the scientific name?
Of
Or
the
it's just
sea spider,
Yeah, or
it's Colossendice.
Cola.
Yeah, so the bigger group of all sea spiders includes way over 1,000 species, the Pycnogonids or Pycnogonidae. And then within there you have several families, including the giant sea spiders, to which this group belongs to, the Colossendidae, over about 100 or so species, some of which get about 3/4 of a meter or so across, so
Oh my goodness.
Wow.
quite big.
Wow.
At this depth?
Yeah, I mean, there's some shallow water species, although predominantly in deeper water. Yeah, they're quite ubiquitous. The bigger group of the sea spiders, found from pole to pole, but predominantly in deeper water. And because they're primarily a deep water species, we know very little about their life history, about how they reproduce, how long they live, and what they eat. And yeah, they're certainly a very intriguing group.
All right, come on.
Yeah, spiders have a special place in Palauan mythology as well. Yeah, there's a story of a spider demigod, which is basically how natural childbirth was introduced to Palau, or that is as the story goes. So it was a spider demigod who fell in love with a mortal woman. And at that time, there was no natural childbirth. It was all only by a C-section. So usually the mother would die. Yeah. Snail. And then he actually prevented them from performing a C-section on her, and she delivered birth naturally, and then that was the introduction of natural childbirth to Palau. And we have a first childbirth ceremony, so a very special place in Palau society.
When was that? Like, when did that story first come about? Do you know?
Oh, I do not. It would be interesting to try and trace back the origins, or
Yeah.
it's all oral history, so.
Yeah, of course. Wow, that's a super interesting story.
And then a sea snail.
This is beautiful. I don't think we've seen one like this before.
No, I haven't seen one like this before either.
Any idea
Oh,
what kind of sea snail?
I don't know. I'm not very good at these.
Yeah, me neither.
Mollusks.
I mean, we don't have any.
Yeah, it's a gastropod.
Definitely a mollusk. that wins.
Yeah, the texture on the shell is really interesting, and the way to ID snails is by counting the whorls and the way that the opening is on the snail, and like a little bit of the texture as well. And I'm not very familiar. There are so many groups of snails that it's a bit overwhelming. And we don't see a lot of them on these types of dives. So this is kind of a really neat observation.
Can we see how big it is related to the lasers? I kind of missed that.
It's tiny.
Yeah, it's really small.
It's really tiny?
It's less than 10 centimeters.
Okay. Yeah, okay. But it might be like 4 or 5 with a little
Yeah.
tail.
Yeah, this shell is probably 2 inches.
Yeah, I think that's great.
Okay, come on.
Thanks.
Oh, we've seen a spider and a snail.
Mm-hmm.
2 things I think we had not previously seen, so really interesting.
I think we saw a spider the other day, no?
We've been seeing a
But
lot of
maybe
sea
you were
spiders.
filtering.
Yeah.
Yeah.
Oh, probably.
We
It's
definitely
possible.
had some. But that was a good one to be your first one to see.
Mhm.
Yeah.
Yeah,
Yeah, because it had the amphipods.
Yeah,
That was neat.
we've definitely not seen that.
it had its associates. It's
Let's look at that little pink spot.
Yeah, we got a cucumber and an urchin. Let's get the cutie first.
the cucumber.
Alrighty, pushing on the cucumber.
This is a sea cucumber in the family Senelactidae called Paleopodides.
Alright, come on. And did you want to zoom on this as well?
Yeah, let's zoom that too.
Okay, you can push in there.
More zooms for Aaron.
Appreciate
This is
you.
a Liponema. It is a type of anemone called a pom-pom anemone.
Ooh.
It looks like a pom-pom on a hat.
My phone. Yeah.
Okay.
Zoom out.
Yeah, yeah. I love those little pom-pom crabs. They're so cute.
We like the zooms too.
Good. I appreciate you all. More zooming.
Zoom in, zoom in.
Yeah, so that genus there of anemones is characterized by very short tentacles and gives them short and many, gives them that pom-pom appearance. So just as a preview here of what's ahead, so we have about a little under 11 hours of bottom time left on today's dive, which is unfortunately the last of Lebu's Voyage 2. So we'll be coming back up on deck around 2 in the morning in Palau. And then after that, we'll have about a 26-hour transit back to Malacca Harbor. En route, we'll do our final eDNA surface sampling collection to support the ongoing monitoring effort by the Palau International Coral Reef Center. Here, my colleague Else and Greta have been judiciously filtering samples and processing them, working after they spend 8 hours here in the control van, probably spending another 4 hours a day filtering and processing samples in the wet lab. And yeah, very grateful for all their hard work, and then building out this sample collection that they have been securing over the last 2 years, collecting surface samples, and now adding a whole suite of additional surface samples, including in locations that hadn't been collected before, as well as in deeper depths. So first time collecting really at depth and different intervals along the water column. So very interested to see what these samples will tell about the true biodiversity of the Palau National Marine Sanctuary.
Yeah, thanks, Daniel. It'll be interesting to see once everything gets sequenced and processed. And I do believe for this dive we have one extra Niskin because the bottom was less than 2,500 meters. So if we see anything of particular interest, or maybe if we get to the top of the feature, are we planning to go to the top, or we're just traveling along the side?
Yeah, the plan is to get to the very top. And the top of this feature is, now there's in essence 2 topographical highs, if you will, so 2 little tiny cones, and that are intersected by a long ridge that goes over several kilometers across. Yeah, so that high point, I expect there'll be a lot of diversity similar and density similar to what we saw yesterday, so those will be really good places to collect some additional samples.
Yeah, for sure. Oh, so a dead sponge?
Yeah, a dead sponge with a basket star and an Acanthogorgia. And maybe let's zoom in on this unbranched Gorgonian coral.
It's a bamboo.
Push in there.
You're braver than I am, Megan. I'll just wait for my zoom ins too.
Oh, yeah, it looked like a bamboo. It was just kind of a little too fleshy, a little too wiggly.
So you heard it. Fleshly and wiggly.
Yep.
Distinguishing characters.
I don't know. It's really hard to like tell people exactly what I'm looking at. It's because I've seen so many, right? When you zoom in, you can see the characters for sure, but from far away, you start to recognize just the general vibe.
Yeah, no, for sure. Yeah.
But since the polyps are closed, you can see those intertentacular spines sticking out. And then you can also see those black nodes between the long carbonate bones, the internodes. And bamboo corals often have more of a peach kind of color to their polyps. They tend to be, I don't know, a little more little more chunky compared to the primnoids. Like they're not as white, and they're a little more fleshy looking than a primnoid coral that's unbranched. And then also the way that those polyps align from far away is a little less like of a rigid alignment. So hence the wiggly.
The wiggly.
The wiggly description. You know, they're just, they look different.
So was that one internodal?
No, it's unbranched. So
Oh.
there's no branching, so you can't describe it as nodally
Nodal.
branching or internodally branching. Yeah, so the corals, they can actually be any combination of unbranched, internodal branching, nodal branching, or a combination of both internodal and nodal. We've seen that before.
Oh, wow.
And we originally thought it had to be one or the other, and then we found a few corals that were like, we don't want. So, yeah, that little theory got taken down a notch there. But we've been learning a lot about bamboo corals. There are a lot of different species in that group, and they're very difficult to identify visually, and even genetically, we're still working out the different species and clades and groups of these animals.
Yeah, so interesting how in taxonomy it'll be like, this is a steadfast rule, except when it's not.
Yeah, yeah, it's kind of like in English, the I before E except after C, but you know, there's like 100 words that don't follow those rules.
Ooh, what is this, sponge?
Yeah, this is a glass sponge in the family Pheronematidae.
Pheronematidae.
Go for a zoom.
This looks crunchy.
It's actually squishy.
Oh, I missed it. I got it wrong.
Yeah, it would be, this is a very spongy sponge. You can see that lattice work.
And then the red, what are the
Amphipods.
amphipods? Very small ones.
Yeah, very, very small.
Yeah, that's great.
Okay, come back wide.
Thanks, Jake. Thanks, Aaron.
Yeah, back to your comment there about rules and exceptions. I think that it's important to realize that all those rules are human-made constructs to try to make sense of the great beauty of our world. And so those are rules that we created to try to simplify things, and a lot of times it's just showcases that we not fully comprehend what's there. Yeah.
Life is never that simple.
Yeah, and it's, you know, the more you look, the more you learn. The bigger holes you poke into those theories. And it just also has a sense of, yeah, how much data you have. And I when I was in grad school, worked on taxonomy, classifying things, and one of my favorite quotes was from a mentor that he said, basically, if you look hard enough, you will find differences even between genetically identical twins. So yeah, if you see two things and you look hard enough, you'll find differences. But if you start seeing 10,000 things, then you'll start lumping things together more. So yeah, the sampling effort is really important. There's a lot of, even among us humans, there's so many differences between individuals. And it's really, you need to see a lot of them to really try to understand what is truly the species, and even the term species has, there's not one definition that all scientists agree on what a species is. There's different kind of concept of species. Original was the old Greeks. They talked about the morphological species concept. So like you can see two things, and if you measure them, and you'll see differences among them, and they'll look different. So that was kind of the original kind of concept, is you can just measure differences in different traits and see them. And then later on, Ernst Mayr brought up the biological species concept, and that basically is that two things are the same species if they can produce offspring together, if they can reproduce. Now that made it simplified a little bit, but it also created a lot of trouble. You have hybrids, you have different species sometime creating offspring, and then a lot of corals and a lot of things, for example, that's very, very common, is that you have hybrids, a lot of reef fishes too. And now in modern day we use the genetic species concept, is that there's the DNA is what holds the key of how. But the basic problem you have with all these definitions is that really how different do things need to be before you say that they're different. And it's where do you draw the line? And the drawing of that line really is a construct of how many individuals. And as I mentioned, if you see two individuals, it's very easy to say this one's this and this one's the other. But if you start seeing 10 million, you'll see that there's all kinds of shade of grays between the black and the white. And in some cases, you'll see kind of groupings that are good species. But in others, it's really a continuum.
Yeah, thanks, Daniel. Very interesting on sort of the maybe philosophy behind taxonomy and the how we are as humans. We like order and we like rules, and we're trying to apply it to the natural world, and sometimes it doesn't necessarily fit in that. And I do think it's nice that we then, with the scientific method, give ourselves permission to change our minds, even though sometimes it can be annoying to have to then call it something else. But, just updating after you get new information.
Yeah, absolutely. I think that humility is something that's really important. It recognizes that we probably will be wrong at some point. New technology, more information might change what that means, and not being too tied down to what we think is true, and really not losing sight of why we're doing these things, the bigger picture. It's really, whether it's 10 species or 15 species, it's not really worth fighting over that much. The main point of this is that it's a lot of species, and we're losing species because of environment impacts and human impacts, and so it's really important to keep that bigger picture in mind as well.
Yeah, I had taken a class where we talked about how do we determine how many species there are in the world, and even just determining that we're already losing ones that we will never see or never discover because of how small they are or how difficult they are to get to. So these are just our efforts here in the deep ocean, and then we'll continue to add to that library of species that are known to humankind. And of course, they're known to themselves, as May spoke about in her presentation. So just they're here, whether humans see them or not. Ooh, there was sorry. I got excited by the jellyfish.
They're here right now.
It's actually a sea cucumber.
Oh, the sea cucumber. Oh.
Yep. The Pelagothuria.
Ooh.
Wow.
That's beautiful.
It's really nice.
It's so shiny.
Well,
It's got something on it. You see that little red
Yeah.
thing? Yeah, it's odd.
Can we turn the lasers off?
Sure.
Let me know if you want to zoom.
Yeah, you can push it a bit if you have it.
Oh, nicely done.
Wow.
It's got a little hitchhiker.
Who's that?
I think it's
Is
a
it?
little amphipod.
Yeah, it looks like an amphipod.
So do these live only in the water column?
Yeah, these are fully pelagic sea cucumbers.
Interesting.
So they're highly modified for swimming, which is why they have that very jellyfish esque body style. Makes for more efficient swimming. You can glide in the water, kind of act like a parachute.
Alrighty.
Nicely done, Jake.
And then the little fish we saw earlier, that
That was a cyclothony, a bristlemouth fish.
oh, cyclosony. Do you know if they normally get bigger or they're normally
They're very, very small fish. They don't
okay.
get bigger than that, really. But they are the most abundant vertebrate on the planet.
Oh,
They're a midwater fish, so they live exclusively in the water column as well. We do sometimes see them associated with the tops of seamounts, because the seamount is intersecting their depth range. Not because they want to live on the bottom, but the bottom happens to live where they live.
yeah. It's probably swimming along and then, oh, a mountain.
Yeah, it's like, boop. So we often do see them on dives, but not because they've chosen this substrate as their home, but because the substrate happens to be here in their depth range. But the midwater, the biome that, from the surface of the ocean down to the bottom is the largest on the planet. So like that takes up, most of the life on our planet is in the midwater. And that fish is quite abundant in all oceans.
Yeah, thanks, Megan. And that's, I think, one of the things that's really important to emphasize here. I think most people have probably heard the stat that 70% of the surface of our planet is covered by the oceans. That's now common knowledge by a lot of folks, but what most people don't realize that most of that ocean is actually really deep waters. The average depth of the ocean is over 3,000 meters deep. For comparison, the average height of the land is only about 800 meters high. So that means that the ocean is about 4 times deeper on average than the land is high. So there's, of those 70% of the surface, that's also in three-dimensional. Its space is much, much more. And that midwater, the water column, that is really an enormous space that we know virtually nothing about, but that also connects the land to the bottom of the ocean and vice versa, and really important. Includes organisms that move up and down the column, bringing energy and nutrients, important for climate, and so much to learn.
Yeah, and the other thing about the midwater that makes it so vast and also difficult to study is that it's constantly changing and moving, just like the seafloor is. However, when we map the seafloor, we don't expect it to completely change form or have all of the organisms move, because some are sessile and they live in a sort of stationary place, but in the midwater, things are always flowing, and there's not really a given destination, so it can be really time-consuming to do midwater surveys and look in these areas. And unlike what we do here, where we map and find a feature and then go up that feature in a way that would be most conducive for the kind of research that we're interested in doing, this kind of habitat characterization across depths. And so I think the midwaters is in that sense really understudied from a scientific perspective, but there are all sorts of different people interested in it and working on new technologies that are better suited for those kinds of environments. And sometimes we work with Mesobot, which is one vehicle that does a really good job of studying the twilight zone and diurnal vertical migration. But yeah, it's always interesting to go in the blue water down to the seafloor and often not see so much and see a lot of marine snow and blue, and just know that there's so much around us that we just are in this one little position moving down that, you might miss what's going on.
Yeah, absolutely. And we can do midwater surveys with the ROV, but I've noticed that the life does tend to avoid the ROV because of that wire in the midwater. If you watch on some of the sonars, like our fish finding type sonars, you can see that big mass of vertical migrators will kind of avoid the ROV. So that's why we need more vehicles like Mesobot that are autonomous and could be part of the environment a little bit more to survey this like very vast environment of the midwater.
Yeah, it's definitely interesting to see how technologies there's whole worlds of kind of biomimicry in technological development to be more blended into a space, so you're less disturbing it in order to study it more like the inhabitants of the space, of the organisms that live there.
Like the octopus camera.
Exactly.
That was so adorable.
Yeah.
Wait, did they put a camera on an octopus?
No, they made a camera that looked like an octopus.
Yeah.
Oh,
No.
okay.
There's no GoPro octopus situation happening.
Honestly, you could probably give a GoPro to an octopus and they would take some great footage.
They're very intelligent.
Yeah.
Though they might take your GoPro apart.
Yeah.
Your GoPro might just blend into an octopus suddenly. Or the octopus might look like a camera.
You might never get your GoPro back from the octopus.
Yeah, Google is just coming up with octopi holding cameras, so.
They've also made, I think, a couple cameras that look like coconut crabs so they could better study the coconut crabs.
That's interesting. Huh?
Yeah, can we look at that white coral, Jake?
The little one?
It was off to the right.
Yeah, off screen now.
Push in there.
Kind of looks like Pleurocorallium kishinoye. But it could also be Paragorgia of some kind. But I'm leaning more towards Pleurocorallium.
That's all the zoom I have.
Are those also amphipods?
Yeah, there's a few amphipods, and then there's a little hermit crab as well.
Oh yeah, on the left.
Mm-hmm.
Yeah. Mm.
All right, I think that's good.
Okay. You can come on.
Is that another piece of wood?
I think it might be a dead sponge.
Oh, it's a dead sponge.
Yeah, I think so too. Let's take a look at this cucumber.
You think that's a cucumber?
That looks like a piece of wood.
That looks more like a piece of wood.
I was brave.
But it could be a cucumber. I don't know. Maybe it is a cucumber.
You can push in there.
It's okay,
It's
I was
odd
brave
looking.
about the dead sponge, so we're 2 for 2.
A wooden
Yeah.
cucumber.
Yeah,
What?
stand corrected. Yeah,
Yeah.
Benthic wood.
all right.
Benthic.
Alright.
It looks like a shallow water cucumber.
Yeah, I was gonna say it looks like
Like
what
a-
I'm used to seeing.
Yeah. So, but generally, our deep water cucumbers tend to be a little more flamboyantly colored.
So how long does wood last at this depth?
It can take a while to be fully consumed. A lot of animals will feed on it, but it needs to be broken down by bacteria, and so yeah, it can last a little while.
Yeah, and what I always found fascinating is that there's whole assemblages, whole groups of animals that are just specialized, deep sea creatures that are just specialized on feeding on both wood falls and even whale falls. So they just basically, once they find these things, they will latch onto them and eat there for a really long time. But just very interesting to have an organism that is their main source of nutrition is not created in the deep sea, it has to come from far away. And as you can see, these pieces of wood are not very common, so it's a highly patchy environment of their food. So how do they make it from one piece of wood to the next? Yeah, it's just a fascinating kind of problem to have, that if you find your food, you can be there for a really long time, but then it might be very difficult to get your next meal. And what kind of life history strategy would succeed in that kind of environment? And that's just, yeah, something very interesting about just the reproductive biology of a lot of these organisms. So the few species where we're actually, few invertebrate where we, deep water species where we have information of their reproduction is, you have typically a bimodal kind of way of reproducing, is that you have a lot of larvae that settle very close to the adult, close to the parents, presumably because the parents are in a good environment, so it's good to have the kids close to there. But there's also some larvae that disperse very, very, very long, and having both of these strategies at the same time is a good thing for, for the long-term survival of a species, so that you're both, close to the good, but also not stay always in a place.
What is it, Jake?
Cascale.
Yep, little casquille. We've been seeing a lot of these ones. I don't think we have a full ID on this, but I know Jeff will help us with these more
Um,
as we get to annotating.
Bazazetus from
Bazazetus,
Jeff.
yeah.
Thanks, Jeff.
The eye looks too big for Bazazetus.
Okay.
Oh,
I'm just used to them having really
Nope.
small eyes.
And courtesy of Ken Sulak, the warning for the root, um, the name Basilzetus, basically, in the English translator would be a fat monster with a compressed body,
No.
and that's where those words came from. So yeah, a lot of
oh my
the
goodness.
Latin and Greek names of these species that sound very foreign to us. They do are descriptive terms to try to tell us something about the appearance of these animals.
Right.
And you can read all about this here probably today or tomorrow in a new blog that will be posted on our website, just describing some of the origins for these names.
I'm really looking forward to this vlog. You're promoting it very well.
Yeah, me too.
Well, as a non-fish biologist, it really helped me learn a lot of the names,
Yeah.
and it does, it's these names sound so like foreign, and then but when you try to you break it down and then the meaning, and it actually has it'll stick with you a little bit longer.
Yeah.
You know the story behind why they were named. And
You're back. How was your interaction?
Oh, it was good. They keep calling me principal, like I still work. But yeah, they had a lot of questions about, and it was just good to kind of walk them through everything. Show them some live feed and what's going on. But yeah,
How old were these kids?
so it's the combination of all of them. So they're from kinder to 8th grade. Well, 7th grade, because there is an 8th grade this year.
Oh, wow.
Yeah, all 11 of them.
Oh, 11.
Yeah. So the island I'm from is pretty far, Kayakala, and I think because of our um, how far we are, they haven't closed our school because there's no transport of bus to take us to another school. So we've managed to have 11 students, and it's also a challenge because then staff is limited. They kind of limit our hiring process, so we're forced to do multi-grades.
Okay.
Yeah, but yeah, it was really, really fun. And
So when you hit 8th grade, then do you go to...
yeah, so after 8th grade, they would go to high school in Palau. They do have a new program that they're doing online schools for the outer-lying states. But most of our students that did it in the beginning, they're so into sports, so they end up going back to Koror so they can do practices and join the sports events.
Okay.
Yeah. But I did an interaction with Angar Elementary School earlier as well, and there were some high school students with them. That's also an outlying school. So it's a great opportunity to keep the kids within the island. But then, they miss out on those sports activities such as basketball, volleyball, competition. So a lot of our kids started the first year, and then the second year they're like, oh, we're going to go to Koror. I'm like, all right. But yeah, very interesting, fun.
That's cool. Are there any little future marine biologists in there?
Yeah, I hope so.
can we take a quick look at that white
Yeah, I can push in there.
What do you think, Megan? Is this, yeah, same thing? It's
Yeah, it looks like Paragorgia.
Yeah.
The rounded kind of retract polyps look like
Oh,
Paragorgia, but then the ones. But is it? Yeah. There's a cup coral that fell over. Yeah.
Yeah, that's great.
all right,
Thanks, Jake.
come on.
Yeah, it looks like that is a somewhat common here on this boulder. There's several colonies all around. And, speaking of, I was talking a little bit about reproduction. A lot of corals and sponges and other invertebrates, in addition to the sexual reproduction, they also have asexual reproduction. So you could have a fragment of a coral breaking off, and that fragment, if in the right environment, could reattach and then build a new colony. And so that's another strategy for reproduction over evolutionary timescale. That is a less of a good strategy for reproduction, because it prevents basically every one of those fragments is identical to their parents, so it doesn't give it ability to change over time. But yeah, you have a lot of organisms which are able to do that, and particularly in these very high flow environments where you have things that can break off. That's a good strategy to be able to reattach and then create new. And in particular in the deep sea where you have as you can see, food is lower, and it's important to have many different strategies to ensure the long-term survival when conditions are harsh. And yeah, we just had one of our scientists ashore. Thank you, Asako Matsumoto, um, expert in a lot of these deep water gorgonians. Also thinks it's pluricorallium. Thank you, Asako.
It's a nice little under ledge
Yeah, it's pretty
crew.
neat.
Yeah.
Just hanging out.
Yeah, that's what I'm saying.
I see what you did
Can
there.
we zoom in?
Sure. You can push in.
Huh.
Anemone,
Sponge.
Sponge, yeah, there's a little cup
sponge,
coral,
urchin.
Korean. Yeah. Oh, there's a Ispidodiadema type of urchin. There's a dema sponge and
Inside? The white one?
yeah, see that the brown is
Oh.
actually a live sponge, but it's a dema sponge.
Oh.
Probably in the family Geotidae. And then there's the white crinkly one in the background. That's a Korea.
This is the spot.
Yeah. The hangout spot.
Yeah.
Sometimes it's good to look under the hood. So it looks like we are just about at waypoint 3. So covered.
we
Waypoint
passed.
4.
Yeah, we're like halfway to waypoint 4.
Oh yeah, thanks. So we're about, yeah. Can you give us a range approximately, Megan? On
Sure.
Yeah.
136 meters.
Perfect. All right, that's helpful. Thanks. Yeah, that stretch from waypoint 4 up to the top should be very, very exciting.
So the top is around 800 meters or so.
Yeah.
770 to 840 or 820, and
Does
so
that
I
shrimp
guess.
have a backpack?
Ooh, that would be cool. So I guess when we come back on watch, we'll still have a little bit of time on the top.
No,
No backpack.
no backpack. It just was a trick of the light.
Yeah, they'll save hopefully the very last for us too. Yeah, we'll have to probably pull off bottom around 1:30 a.m. to make it to the top, and yeah, so that last hour and a half
Nice.
should hopefully be at the top of the ridge. And it's a very somewhat narrow feature, and I expect to see a lot of abundance of different organisms up there.
Yeah, that'll be really exciting.
So yeah, we have exactly 12 hours remaining in this dive. And have about a little over 2 kilometers to go if we were to make all of our waypoints. So we'll try to balance out covering some ground, but also getting some good close-ups and still photographs, as well as samples of anything that might be of scientific interest. Let's
Ooh,
take a look at
Astinactus. I like these sea stars. They're really fun.
Has 8
All right,
legs.
Max. Push in there for a zoom.
I've always just wanted to touch it. It looks like it would have a really good texture.
What are these called again?
Astinactus.
Astinactus.
All right, come back wide.
Chris, so Jeff says that Chris says Solengaster?
Solenogastries.
Solenogaster. Huh? I mean, if it's Chris Small, then he knows better than me. It's another neat sea star.
small fish on the left. Looks like Basazitis. Could be a rat tail though.
What did you say Basazitis means? Short, short, squat monster.
Alright, Max, you can push in there.
It was a fat monster with a compressed body.
The technical term.
Yeah.
Well, this fish has a small dorsal fin, so it is not a Basazitis.
Oh.
This is a macurrid, a rat tail fish.
Might be a Bathygatid. It's got that second dorsal too.
Yeah, could be. You can see those long filaments on the pectorals as well.
Hmm.
It's a very little one.
Alright, come on.
I'm going to make a prediction here that our video engineer will be studying a lot of fishes in the near future. And
That's the hope.
Yeah, one of these days Max is going to sit in all the seats too.
That would be pretty cool. All right, giving it back to Aaron.
Bye, Max.
Thank you.
What is this critter?
This is a little baby cutthroat eel.
Oh.
Looks like a juvenile.
Oh my goodness.
Yeah, if you zoom in, you'll be able to see
tiny.
right through its
Push in there.
body.
Yep.
Its little beady eyes.
Yeah,
It's so
it's a little
cute
baby.
to have such
yeah.
a
To
vicious
see the
sounding name.
intestines.
Yeah. Well, they're not called cutthroat because
know.
they're fishes.
I know, it's just,
They just have, unfortunate gill placement.
Huh.
Stalked crinoid.
So I was on, it was the cruise with you, Daniel, in 2019 when we had a whole bunch of squids off the east coast of the U.S. and there was a crab and a cutthroat eel fighting over a piece of squid.
yeah, I remember that.
Yeah, and then this other science lead, Jeff, and I made a very silly joke. I was like, these eels can be pretty hardcore when it comes to feeding.
Hang on, wait for your punchline.
Would you say they're cutthroat, Megan? Yeah, that was how it went.
Oh, what's the little orange?
Oh, it was a squat lobster.
Wow.
It
Swimming
started swimming.
away.
Yeah, it was like swimming backwards. I love when they do that, the little
Oh.
scoot.
Oh, it's like going like tail first.
Yeah, it goes tail first, like a lobster. That's their quick swimming mechanism.
is that a dead sponge?
Small fish.
Fishy, getting a little fishy.
That's
Yeah,
right.
getting some more fish going on. That's always a good sign.
Push in on this fish.
Yep. Another little macurrid, rat tail fish. This
Oh,
one is not
Jeff.
the same. It's not a Bathygatid.
Jeff wants to do a collection.
Wants to slurp it.
What do you
Okay.
think
All right,
about
how
that?
you feeling?
It's small enough. Yeah, that's for sure. Alright, come on.
Yeah, I think also it looked like a kumba, which also I learned a little bit about, that is an anagram paying tribute to a journal, the United Kingdom United Kingdom Marine Biological Association and their research vessel, and they've had a journal too, and that vessel collecting the type specimen of this species.
Yeah, that's fine. We don't have any collections yet. It looks like there's a little bit of sediment in there, but it's no problem.
Nah, that'll be fine.
So this fish should really be named Ukma, not Kumba.
Yeah, but that just doesn't roll off the tongue as well. And Kumba is such a cute name for such a cute fish.
Okay.
Oh no.
Oh, wow.
Stop.
Swift.
Right where I wanted it.
It's still
Right
there.
on top. Yeah. Top of the screen, yeah.
It looks like an oversized tadpole
Yeah.
from far away.
So, oh, left.
Oh, I see it.
Oh, there he is.
That's it.
Go.
Well, that's no help.
This is just.
I don't know.
Yeah.
Kind of swimmed into the vehicle.
Yep.
It's not down there. I think it swam off.
I think we'll unfortunately probably have to move on, but that was a very valiant effort, and thanks so much for trying.
Yeah. Did you all hear about the sponge that jumped into the bio box yesterday?
We
Really?
jumped in?
So
Sponges can jump.
on a previous watch, not ours, there was a sponge collection happening, and there was a nearby sponge next to the sponge that we were collecting that independently kind of came off and
Fell into the
beautifully fell right into the bio box with its sponge companion.
Oh,
Like,
wow.
don't leave me.
So we got a 2-for-1. But yeah, it was kind of interesting to hear the watch team, the science team from that watch were completely stunned because it wasn't really touched or knocked. It just kind of seemed to fall off somehow on its
Seemed
own.
to jump.
But
It just really wanted to come home with
Yeah.
us.
yeah, sometimes we get those opportunistic sampling moments, and sometimes the fish swim away, and
Yeah.
it's all part of the process.
Fish or the sea cucumber.
Yeah.
What are those yellow objects?
Those are encrusting Demis sponges.
Oh, I thought they were our Gucci.
Do you want to look at them? Or?
No,
Gucci.
well, we can look at them, but they're not the Gucci Somnia.
Okay.
Gucci Samia.
We're too deep for that, I think.
Yeah. Wishful thinking.
Oh,
Yeah.
they're a lake shallower depth.
Yeah, we should just, I mean, we don't have to that one, but we should try to get just one zoom later.
Yeah.
Yeah,
think they're abundant
all,
enough.
Yeah, they're all
Yeah.
over the rocks here.
I love how you were just ready to say the answer. You like didn't even have to think. It's so natural.
I could feel it. I could feel he wanted to know.
What's that pink thing?
Pink
It's a heavy
coral.
corallium.
Did it fall over?
No, it's just little.
Okay. Oh, no, I see.
So for the sponge, under the sample type, is it volunteer?
Ally? No.
Method of collection.
It
Volunteer.
willingly came.
We paused for a moment in the lab. We have a category for subsample or associate or kind of an additional sample to the
Right.
original intended sample. So we got one of those since it was at the same site in the same box. And
Yeah.
Let's do a snap zoom on.
Yeah, oh, and there's one of those yellow sponges up there too.
Perfect,
Push
yeah.
it there.
So this is a primnoid coral. Looks like Calyptrophora wyveli. And it has a snake star wrapped in the branches.
Wonderful.
A little urchin to the left.
Yeah.
yeah.
Alright.
Thanks, Jake. If we can catch, yeah, just quick that yellow sponge on the rock just for documentation purposes. There you go. Perfect. Thank you.
Nice.
Wonderful. Thank you.
Okay.
Yeah, I want to look at that squat lobster. I don't think we've looked at one of those yet. Yeah, that's a good one.
It's a little more,
Push
it's
it
a
there.
little more substantial.
Oh.
Oh. Wow.
Well, we got a good view of the swim. That's a little exciting.
Another primnoid coral and an acanthogorgid coral.
Yeah, that one looks like Acanthogorgia.
Is that the one, the formerly plexaurid?
No, no,
Oh,
it's the
different.
original flavor.
Oh. Oh, so the former plexaurid was added.
Yeah, the ones that were formerly Plexauridae have been moved into Acanthogorgiidae.
Ah.
And
Along with those.
Yeah, along with those. So the original Acanthogorgia, they can't fully retract their polyps because they have a bunch of spines on them. And so they tend to look really fluffy,
Mm.
and they tend to have like a denser polyp um, distribution along the coral, as well as the color tends to be a little lighter yellow.
And then
They
the,
just look fuzzy.
yeah.
That's usually how I tell, and they're very, very flexible. So as we kind of drifted by, you could see it moving in the current really easily.
Yeah, it's maybe a
Yeah,
quick snap zoom on that urchin.
it's a pretty one.
Yep, we can push in there.
And so this is an urchin in the family Echinothuridae. They're characterized by these thin spines, and they're known as the pancake urchins. And they often have these little boots on the lower spines that help them walk across sediment.
The black part
No, not the black parts.
on the tip?
The lower spines. You see
Oh,
how they have
okay.
like little white tips?
Oh,
They're like these little shoes. They wear little boots.
wow.
Kind of flares out the ends of those walking spines to help them move. The Tromicusoma, the big round purple ones, have like really nice boots, but this one has very small boots.
Okay.
Yeah, and I got a picture here of the deep sea animal guide. Looks similar to a Sparrosomatinae
Yep,
species from a Holland and Baker.
I would agree with that. Yeah, we used to call them Spirosoma, but then we learned that there are actually a number of different genera that it could be, and it's impossible to tell from imagery, so we've been only going to the subfamily. And there's a nice persingent sea star. So these sea stars are filter feeders. They keep their arms up into the water column and grab things to eat from the water. And so when you see them in that posture, they're actively feeding.
And when they're not feeding, it just comes down?
Yep. Then they'll relax.
Brazinga.
Bazinga. Kind of sounds like bazinga. I feel like we should just start saying that, just a bazinga.
Let's maybe do a snap zoom on.
Is that
Yeah,
a
it's a
cookie
little
star?
cookie star, yeah.
Cookie star.
So we had a pancake urchin, we had a cookie star.
Yeah, we're ready for tea time.
There, you can push in there.
Oh, this one has little scale worms on it. Those little yellow animals are scale worms.
Ah. So those are, what, eating it?
No, no, they're just associated.
Associates? Okay.
Yeah.
It's really nice how they have that, the outer layer kind of look. Yeah.
Yeah, I like the texture of these stars.
Yeah,
Okay, come on.
they kind of look like stingray texture or something.
Yeah.
Stingray.
Tall chrysogorgid.
Bridge now. Hey, how's it going? I see you changing heading. Okay. Cool. Sounds good. I felt the bow thruster getting a little.
Was a big bathypathes, black coral. So yeah, all the black corals there.
A
are
black
always,
coral?
it's off frame now.
Oh, I was like, I don't see it.
This
I
is
was
the
looking at
chrysogorgid.
the DP screen. Yeah, and more of that same furriid sponge that we've been seeing consistently at these depths throughout Palau. Oh, it's raining again.
And thanks here to Ken Sulak sharing another fun fact here in the chat. That those large pancake urchins that we just zoom in, that they often provide a shelter for juvenile fishes. A lot of small cuff eels are typically seen underneath them, and some very small synapobranchid eels. So
Yeah, I've seen that before. It's super adorable.
Push in there.
We saw that quite a bit, actually, I think, on the East Coast when we were there in 2019.
Oh.
And there was a few observations on the capstone cruises as well.
Guys.
Between 2015 and 2017.
Can you hear me?
Yep.
Yeah.
Ah, sorry. Yeah, Jeff would really like to collect the rat tail. He just had to leave for a second. Sorry.
Okay.
Sorry to interrupt. I thought it was pretty urgent.
Not surprised.
Shall I come wide?
Sure.
Where's the rat tail?
Uh,
I don't see it anymore.
that's not a rat tail?
No, that's a
Oh.
eel.
Oh, yeah.
I never saw it.
I didn't see a rat tail.
there's a question here in the chat that's pretty interesting. Can there be too many associates, like to a point where it becomes a problem for the host?
That's a good question.
Yeah, that's a great question. I would have to say yes. You know, if you're so covered in associates that it keeps you from being able to feed efficiently, you could be essentially smothered. Um, what that threshold is going to be highly variable depending on, you know, the species. And a lot of times, like, especially when we've seen like those stalk sponges and things like that, and you have a lot of associates along the stalk, then the stalk isn't like the feeding portion, and so having associates there is not detrimental at all. And a lot of times the corals will have like the snake stars and stuff on the coral, and those could actually be beneficial to the animal by keeping the coral clean and don't, you know, disturb that coral's ability to feed. But we've definitely seen cases where an animal has like so many crinoids and so many brittle stars, and that's usually an indicator of that animal like either dying or have passed. So yeah, I think there is a threshold there where it's gonna not be a positive experience for the animal, but we don't often see that. But also we don't have a lot of data to indicate where is it negatively impacting the animal or not, because a lot of times, especially for this expedition, this is the first time we've seen any of these places, and we're documenting these animals for the very first time in this area. And I do know a lot of the animals because I've done explorations in other parts of the Pacific, and there are similarities, but as you've seen, there has been a lot of things that I don't know, and there's a lot of new stuff to discover here.
Thank you.
Yeah, and a couple more things. Megan really touched on it very nicely. That it's a bit of a chicken and egg thing. Are the many associates there because the coral's unhealthy, or are the associates making it unhealthy? And basically, a lot of times it's a function is once you have parts of the tissue that die, then other things take hold of it. So it's already not being healthy. So a lot of corals, for example, they have many mechanisms for cleaning their tissues constantly because there's a lot of things settling on them. So they have, now if you were to look under a microscope and look at all the polyps, all the of a colony, you'll have every so often different types of polyps with different types of tentacles. They're called sweeper tentacles, and they're much, much longer than a normal tentacles, and they just use those to clean their body and basically just clean themselves. And yeah, and we've seen this here is like once you see dead colonies of things, the sponges, corals, they're typically covered in hydroids and many other things. And yeah, it's usually just as once the coral or the organism is no longer able to clean itself, then everything else will take over. But yeah, there's all kinds of things from very opportunistic association to the beneficial ones or the kind of neutral ones.
Yeah, thank you.
Alright, we're getting close to waypoint 4, and that's where it's really going to start getting interesting, I think. That next transect over to the first of the topographical high points.
Okay. Para an eel.
Yeah.
Push in there.
Yep, this is a cutthroat eel, Synaphobranchus.
Is that a...
All right.
And we can let it go.
Yeah, that's great. Thank you.
Are these dead corals or...
That looks like some dead sponge, and
Oh,
it could be some wood.
and wood, okay.
Ooh, ctenophore.
It's a nice color. What is it?
It's a ctenophore.
Ctenophore. You related to Jennifer?
They are a type of jelly, but they are not like a jellyfish. They're in their own phylum. And so that one, I believe, is a cyctipid ctenophore. The common name is comb
You
jelly.
can push in there.
Huh. That's so nice. Nice color.
Excellent Victor Gorgia and the Primnoid.
Such a pretty color every time I see it.
Yeah, this looks like probably an arella primnoid coral. There's also hemicorallium and Faria.
Is that a hole in a rock?
Yeah.
Yeah.
What is this? What are these legs? Oh, that's the brittle star?
Yeah.
And the sponge just below, just out of view, is that similar to the one we collected yesterday? It's just lower on the rock. Yeah, right there.
From the bottom?
Yeah.
Oh.
It looks kind of like one we collected yesterday. A lot going on in this little boulder.
Yeah, just a little rock and lots of life.
There you go on.
I think this experience has given me a lot of appreciation to sponges and corals. And I think my next time spearfishing, I'm gonna